Welcome to the second day of the Fish Predation workshop. And again, our focus is on the state of the science and not the management questions, but what are those science questions consistent with the charge that we gave the panel. And before we get started here, I just want to thank our panel again. I know Gary, Tim, Brett, Jessica, Nancy, and Todd have been doing a lot of work. They took the information yesterday and have been working on digesting it well into the evening, up early this morning and working together on this. They're going to give us their initial feedback of what they're seeing. This does not mean it's the final. They've kind of outlined a few things they want to go through, but they're going to share some of that with us. After today, they're going to meet again and then work from their various locations over the next 6 to 8 weeks and put together a report which will be submitted and we will be posting on the website. So you should be able to look at that probably early to mid-September if everything goes right. We have some final copy edit reviews to do on it. We're not going to adjust the content or anything, just typo kind of stuff to make sure it's publishable. With that, Gary Grossman, who agreed to be the chair of our panel, is going to give you a readout. And again, if you don't recall from yesterday, Gary has authored over 100 scientific publications on freshwater and marine fish communities and populations, habitat selection behavior, including predator effects. He's a professor of animal ecology in the Warren School of Forestry and Natural Resources at the University of Georgia. And thanks for bringing the humidity today.
There isn't any humidity.
This is not humidity, yeah. And fortunately for us as a chair, he's also on the editorial board for freshwater biology and ecology of freshwater fish and writes a bi-monthly column for American Angler magazine. So appreciate it, Gary. So what we'll do is Gary will give you their initial readout, and then we will take a short break, and then we'll come back and we'll have another opportunity for public comment. Gary?
Okay.
Thanks.
Thank you, Greg. I hate microphones, so I'm going to push this away. Can everybody hear me okay in the back? Oh, well, that's why I didn't want to use it. All right. Everybody hear this okay? Not too loud, not too soft? All right, well, I'd like to thank all of you, the presenters and the organizers, and especially the public who have shown a significant enough interest to be here today. I want to reemphasize what Greg said, that this is a very preliminary report back to you. Obviously, we've been presented with a massive amount of information, and it takes time to integrate all that in a synthetic way. So what I'm going to talk about today are just our preliminary, our meaning the committee's preliminary observations regarding the information that we've read and saw yesterday from the presenters. Which one of the buttons goes forward? Because none of them seem to. The top, where the pointer is? To the right. Well, the right's not doing anything. Oh, there's probably nothing plugged in. Okay, you know what? I can just do it like that. Okay. All right, so this slide just essentially gives a little bit of information regarding the background of the workshop. So the workshop and all of the activities that went into it were a result of an interagency framework. All the relevant agencies got together. They took stakeholder input on the development of the charges, panel selection. The panel members are all a variety of experts in different areas that are relevant to the task at hand. There was stakeholder input and agency input into the readings and the presentations, and the panel's charge was to address a suite of questions regarding the effects of fish predation on salmonids in the Central Valley. Okay, there is also an opportunity besides the question and answer period today There's a website where you can have additional public commentary. This is just a very basic schematic of the predation process for fishes, or really any sort of predator-prey interaction, just to sort of give you, to ground you in the types of behavioral factors and also population factors that are relevant to the predation process. So a predator enters an environment and searches and perhaps encounters a prey. It decides whether or not to pursue that prey and attack it. If the attack is successful, it has to handle that prey, turn it around in its mouth so it can swallow it. And then finally, it has to consume the prey. And all of these aspects have behavioral components, but they're also functions of both prey and predator abundance, spatial and temporal overlap of both predator and prey, and these things enter or affect the encounter rate, the rate at which predators encounter prey. So the first question that we were tasked with addressing is this one right here. What is the ecological context of predation by fish on Central Valley salmonids, and what can be learned from other systems to inform our understanding of this process? It's kind of boring to put whole sentences up there, but these are the exact charges that we were tasked with, and I want everybody to know exactly what they say. Well, probably everyone in this room understands that the delta and bay system is extremely complex, and the committee or the panel tried to capture in this slide some of the relevant factors that influence predation within the system. So physical and chemical factors that affect the predation process or have an effect on the Bay Delta environment are things such as turbidity, submerged aquatic vegetation, temperature, salinity. It's a whole suite of factors that most of you are probably familiar with. Another factor that affects this predator-prey interaction are changes in the predator assemblage that have occurred over time. So once again, as you probably know, or you may or may not know, the native predators that dominated the Bay Delta system have mostly been replaced by non-native predators. So striped bass, largemouth bass, spotted bass, white catfish, channel catfish. All of these species are not native to the Bay Delta system. They are species that do not have a shared evolutionary history with the native fishes or with the native salmonids. And these are all things that are going to affect this predator-prey interaction. Okay, and then we have the characteristics of the prey. So one of the things that's happened in the Bay Delta system is that most of the native macroinvertebrates, which serve as the prey base for both young and adult salmonids, and really all young and adult fishes, has shifted from a native suite to a non-native suite. Things such as the overbite clam, the loss of Neomysis mercedes, and replacement of native microcrustaceans by invasive microcrustaceans. Several other factors that affect the specific predator-prey interaction between predators and salmonids. We have low abundances of smolts now. We have the possibility for depensatory mortality, and for those of you that aren't population dynamics experts, depensatory mortality is an odd situation where as populations grow smaller and smaller, events like predation have a larger and larger impact. So normally, as a fisheries biologist or as anybody that's worked with fisheries, all of fisheries management is based on the idea of compensatory mortality. That being that when a percentage of the population is removed, demographic factors such as fecundity or growth, those things will respond. You'll get higher, as the population decreases, you'll get higher growth, you'll get higher fecundity, And that's what allows the population to persist in the face of exploitation. So we've learned over time, however, that there is the possibility for depensatory mortality when populations become very small. And typically, the biggest issue we worry about with depensatory mortality is predation. And so depensatory predation is the case where, as the population is being driven down, 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 instead of the predators taking a smaller amount of the population, as you would think with compensation, they actually take a larger and larger portion. So this can become a really important process for small population sizes, and especially things that are small enough to be considered endangered or threatened. Okay. One of our first charges was to examine the literature with respect to what we know about predator-prey interactions and movements of salmonids from other systems, what we can take from those other systems and apply to the situation that obtains today in the Bay-Delta system. And once again, it's probably not a surprise to anyone that the Delta really is a unique ecosystem. And that means that what we might know about salmonid predation in the Columbia or the Fraser or the Willamette River may have very limited applicability to the Delta, just because those are very different systems. And just think of the physical habitat. Those rivers that I just mentioned are all linear systems, which are going to have very different dynamics than the Delta, which has flow coming in from two directions, is tidally influenced, so on and so forth. So unfortunately, when we try to address this question and look at what's known from other systems, we find that much of it doesn't really apply to the Delta. The second factor, which we can take from other studies, is that as far as predators go, salmonids are highly desirable prey. I mean, basically any predator that can catch a salmonid tries to, because they're nice little packets of energy swimming around. They have soft rays instead of spines, so they're easy to swallow. So in terms of looking at what predators in the Delta might be eating or might want to eat, salmonids are right there up at the top of the list. There have been a number, one of the things that has come up in our readings and has come up at least tangentially in the presentations is the idea that if predation is significant, then perhaps we should remove those predators, and perhaps that will result in a concomitant increase in the prey population. And in reviewing the literature, what's clear, and I'll also point out that there are very few of these removal experiments that are actually done well enough to reach a strong conclusion about the effects on the prey. But what results we do have, even from good studies, indicate that the results of removal are highly unpredictable, that we can't go in there and say all we have to do in terms of management is get these predators out and the salmonids will be fine. The science just isn't there yet. And it's not clear if the science will ever be there, because as you might imagine, the logistics of removing large mobile predators, which is what we're talking about, are very difficult if one attempts to do it in any sort of habitat rather than a very small reach. Another lesson from the literature that is strikingly clear is that predator-prey interactions are context dependent. And so what we scientists mean when we say something is context dependent, it is merely that a striped bass and a trout in the Hudson River, that predator-prey interaction might be totally different than the predator-prey interaction between a striped bass in the Old River and a steelhead in the Old River. So there are a lot of factors that affect these interactions. In terms of just general comments about populations that are in trouble, a survey of the literature in North America shows quite clearly that there are two main causes in the loss of native species. That is habitat loss or habitat alteration and the introduction of invasive species. You all know that those are two major factors, two major changes that have occurred in the Delta. And I'm not a Delta scientist, but if memory serves me correctly, there are actually more invasive species now in the Delta than there are native species. Am I speaking in the mic loud enough so you're getting this? Yes? No? Okay. All right. Another important fact that was gleaned from the literature is the fact that in terms of population dynamics, fish populations have what are known as critical stages. In other words, survivorship from yolk fry to swim-up fry in salmonids might be a critical stage in some populations, meaning that mortality at that stage is translated upward in all the way up through final population size. So this means, and unfortunately we don't know what these critical stages are for most populations, but the point here is that determining what regulates population size is not a simple process. You can spend a lot of time looking at the wrong stages of a population and never really identify what the critical stage is. So these are really just all to link what's known from the literature to the specific situation of fish predation versus salmonids in the Delta. All right, here are our charges 2 and 3. What do the available data and analyses tell us about population level effects of fish predators on salmonids in the Central Valley, and what do we agree upon, or what do the scientists who have produced these works seem to agree upon, and what don't they agree upon, and what are the key uncertainties? And so this is a bit of a busy slide here, but let's start out with what we do know, all right? There is no doubt that predation on salmonids occurs in the Delta and in the Bay probably, and out in the ocean as well. We know that. One thing that is noteworthy is much of what we do know is based on behavior and studies of hatchery fish. And this could have actually gone down to the don't know as well as up in the know. So we know a lot about the behavior of hatchery fish, but we don't know how extrapolable those studies are to wild fish. And there are a variety of reasons for that. Of course, some of the runs are listed, some are not. Wild fish are hard to catch. So there are a lot of logistical reasons. But the literature shows that in many areas, and even specifically in the area of predator-prey interactions, that hatchery fish behave differently from wild fish. And typically hatchery fish are more naive, so they suffer higher predation rates than wild fish. And so this poses a bit of a problem when we have a situation where most of what we know about the behavioral interactions of these stocks comes from hatchery fish. We know that survivorship is low for all the stocks in transversing the Delta, especially in comparison to other stocks in other areas. We know that survivorship varies both spatially and temporally. If you're in one part of the Delta, your survivorship may be 0.6. If you're in another part of the Delta, it might be 0.02. So one of the aspects that makes conclusions about these processes difficult is the fact that the Delta is a spatially and temporally heterogeneous environment. Okay, so, and one example of that is we know there are some mortality hotspots. And what we mean by mortality hotspots are just areas where there's very high mortality. Typically, these are associated with man-made structures, alterations of the environment. And one that we heard about yesterday was Clifton Court Forebay. Another that's been described in studies but wasn't really talked about is Red Bluff Diversion Dam. typically when we're concerned about population level effects of predators on prey, we look for statistical relationships. And although there aren't a lot of statistical studies in either the data we were presented with or the presentations, there appears to be no relationship between populations of striped bass and salmon. And there are probably a variety of reasons for that, but it makes our task a little more difficult, given that if there was a very strong relationship between striped bass, and typically we would look for a negative relationship if predation was important, if there was a very strong relationship between striped bass and salmon, then it would give us more information to infer that striped bass. And we have striped bass up here because that's what those are what the data were for, but this could easily be largemouth bass or white catfish. Our charge is fish predation, not just striped bass. So if we found a negative relationship, we'd have greater confidence that there was a strong negative relationship between predation and salmonid populations. But simulation studies show us that there are a lot of predators are out there. They have the capability of eating a lot of prey. Now, that's a different thing from whether they do or not. But they have the capability of consuming, and I think the estimate was 3 times the number of salmonids that the salmonid population size or the smolt population size that's typical in the Delta. All right, so that's what we know from the literature and from some of the presentations. What don't we know? And these are the things that serve to make our life a little more difficult, but also make it a little easier later on in that they allow us to identify future areas of research. The first thing that was striking to the panel is that as far as we could tell, there are no accurate population size estimates for either striped bass, predatory striped bass, predatory largemouth bass, et cetera, nor for the salmonids in the Delta. And that is problematical. Okay, how can you estimate the effects of predation when you don't even know how many predators are there? And I should add that there's a spatial component as well. So we don't know, not only do we not know the total number, but we don't know really how they're distributed within the system. We don't know the effects of tags, and much of what we do know about survivorship and movement of salmonids in the Delta is based on tagged fish. They're hatchery fish mostly, but besides the hatchery fish problem, we don't know what the tagging effect is. Yeah, there are short-term, basically when people tag, and I see some people who presented tagging results in the audience, so you can correct me if I'm wrong, but typically what's been done in the studies that we read is that the fish are tagged, they're put in a holding tank for 6 hours to 24 hours, and then they're released in the system. So we really don't know, because none of these fish are kept for weeks, we really don't know what the long-term effect on tagging is. And it's an important component, and not to get too detailed here, but at least for some of the tagging studies, the size of the tag is above the recommended weight for the size of the fish. And as a consequence, there could be mortality associated with that. Okay, another big unknown is mortality itself. And that is, in many of the studies that we read, a lost tag is considered a predation event. But there are many possible, a lost fish does not necessarily have to have fallen to a predator. It could have died from a variety of reasons, some of which are mentioned here. And we even have the more complex phenomena which was brought up yesterday, that being ultimate and proximate causes. The proximate cause may be that there was a predation event, but the ultimate cause was that the fish was so stressed by a hostile environment that anything could eat it. So changing the predators there may not, or affecting the predators, managing for the predators, may have no impact on the prey population if the ultimate cause was that it's living in a really poor habitat and it can't survive for very long. One of the things we don't know, and I've already touched on this, is that we don't know how salmonid populations will respond to changes in predator populations. We simply don't know, and the literature shows that these sorts of responses are unpredictable. All right, so in trying to answer these charges 2 and 3, our conclusion, or the question is, does fish predation comprise a significant portion of salmonid mortality in the Delta? The available information doesn't really allow us to answer that question. And when we do the final report, we will have a section on where we think future research should go to answer this question more definitively. We will not present a lot of those thoughts in this presentation because we need a longer period of time than 14 hours to think about them. Okay, our next charge is, are there appropriate methods for estimation of predation rates and population effects from the existing data? And from the previous slide, it should be obvious that key bits of information are lacking to really estimate predation rates and their consequent population level effects on salmonids. We do think that historic data should be used in models to generate mechanistic hypotheses, which then are tested via future empirical studies. And I told you we'll get into this in greater detail when the report is issued. So next, what biological and physical factors are likely to affect the impacts of predation on salmonids, and what is understood about the interactions among major factors influencing predation on salmonids, interactions among predators, hydrology, and temperature. These are just a suite of 3 sets of factors that the panel has identified as important to these processes. There are physical factors such as flow, and by flow we mean everything everything water can do, basically. So direction of flow, speed of flow, so on and so forth. Temperature is obvious. Habitat should be obvious, as water quality. With respect to the predators themselves, the factors that affect and interact with these other 2 categories are the species of predator, density of predator, size of the predator, origin, native or non-native, and distribution through the delta. And for the prey, we have a similar suite of characteristics as we do for the predators. And so all of these factors interact, unfortunately, in unpredictable ways, right? They're context dependent as well. Another aspect of the research that's been produced that struck the panel was there seems to be a lack of, even though the Bay-Delta system is highly variable, there are drought years, there are flood years, so on and so forth, a lot of the research is not phrased or not examined within the context of that variability. So, in the case where there's maybe a 1-year study, there seems to be not much of an attempt to take those results and integrate them within the larger framework of environmental variability that we see within the Delta. Okay, have these factors changed over time, and do they vary between the major basins? And of course the answer is yes, right? We had a very nice historical introduction by Larry Brown yesterday, which went through the environmental changes quite thoroughly in the Delta. In the introductory presentation on hydrology, there was fairly clear evidence that there's not one homogeneous delta, but at least in terms of flow and environmental characteristics, probably 3 linked but somewhat distinct, I don't really want to call them basins, but let's call them areas. And that conditions within these basins have changed rather substantially historically. Okay, do these factors vary among the major reaches of the system? Yes, obviously they do. I mean, the predation process in the bay or the ocean is likely very, very different from the predation process in spawning areas and the rivers that constitute spawning areas for wild populations. Unfortunately, little's known about spatial variation, especially when we get to the lower end of the system. We do know, to reiterate again, that there are persistent hotspots of low survival, and I would expand this a little bit. We had talked previously about anthropogenic alterations which had produced predation hotspots such as Clifton Court or the Red Bluff Diversion Dam, but clearly areas like some spots in the Old River, maybe the DCC, these are going to be persistent spots that are problematical perhaps for smolts. Okay, I've already mentioned that we need to think about this rather long question here. All of what we think will be in the final report, which should be available in a couple of months. In viewing the research, one aspect that once again was notable by the panel is that a lot of research in the Delta is conducted by separate organizations for their own specific purpose rather than being integrated into some overarching conceptual framework within which more specific and more important questions could be addressed. And obviously anybody that's taken a experimental methods course, the idea is you build on everything and then you get the big picture. And I'll have the next slide will show that graphically. Another suggestion that we would make is to have some standardization of methods so that if 3 different organizations are doing tagging and release studies of smolts, there's coordination, so at least the results are comparable and the methods will be able to address similar sorts of questions. And so this is just when we look at the data and the papers, what we see are a lot of different patches that somehow have to be integrated into a quilt or a blanket. And what we really need is a blanket with different areas, but something that represents an integrated whole in terms of producing definitive answers that can be used for managers. All right, and I started out thanking everybody, and the panel will end by thanking everybody, and thanks for your time. We'll be glad to answer any questions to the best of our ability.